Good morning students, welcome to my channel MicrobeXacademy. Today we will discuss 10 MCQs on general microbiology. So let's proceed. Coming to question number 1, the most resistant form of microbial life is option A fungal spore, option B bacterial spore, option C mycobacteria, option D non-invasive virus. So could you please guess the answer? The correct answer is bacterial spore. So let's move to the explanation. Coming to the first option, that is fungal spore. We all are aware of that fungal spores are less resistant. Regarding option B, bacterial spore. Bacterial spore contains dipicolinic acid and thick cortex which provide resistance against heat, chemicals and radiation. Regarding option C, mycobacteria. Mycobacterial cell wall contains mycolic acid which confers resistance against acid but not against extremes of heat. Regarding option D, non-invasive virus, as we all know that they are not as hardy as bacterial spore. Coming to second question, the indicator organism used for autoclave quality control is option A Bacillus subtilis, option B Clostridium perfringens, option C Geobacillus thermophilus, and option D Micrococcus luteus. So please guess the answer. What is the correct answer? The correct answer is option C, that is Geobacillus steatothermophilus. So move to the explanation. Coming to option A, Bacillus subtilis. Bacillus subtilis is used for quality control of dry heat procedure like hot air oven. Option B, Clostridium perfringens. It is not used for any sterilization quality control. Same for option D, that is Micrococcus luteus. Regarding option C, Geobacillus steatothermophilus, this is a thermophilic organism and is a spore former, and it is ideal for autoclave testing, the quality control of autoclave testing. Coming to question number 3, dry heat sterilization acts primarily by option A oxidation of cellular components, option B denaturation of proteins, option C disruption of lipid envelope, and option D DNA cross-linking. Actually the question is asking about the mechanism of action of dry heat. So guess the answer. The correct answer is oxidation of cellular components, that is option A. So let's move to the explanation. Regarding option A, oxidation of cellular components, actually this is the modus operandi of dry heat which causes oxidative damage of cells. Regarding option B, denaturation of proteins, this is the mechanism of action of moist heat. Regarding option C, disruption of lipid envelope, this is the mode of action of organic solvents like alcohol and detergents. Option D, DNA cross-linking, this is the mode of action of chemical stimulants. Okay. Coming to question 4, ethylene oxide sterilization is mainly used for option A glassware, option B heat labile instruments, option C culture media, option D metal tools. Please guess the answer. The correct answer is heat labile instruments. So let's go through the explanation. So coming to first option, that is glasswares. As we all know, glasswares can be easily autoclaved. Regarding option B, heat labile instruments. As the name suggests, the instruments are heat labile. So we can't apply heat to these instruments, because if we apply heat, then the instruments will get damaged. So to avoid that, we should deploy some other methods for sterilization. So what methods we can apply? One example is gamma sterilization or ethylene oxide sterilization. Option C, culture media. Culture media can be effectively sterilized by application of moist heat. And regarding option D, metal tools. Metal tools can also be sterilized by autoclaving. Coming to question 5, which immunoglobulin is first to rise in primary infection? Option A, IgA. Option B, IgD. Option C, IgE. Option D, IgM. This is a very easy question. You should not take much time to answer this question. Guess quickly. The correct answer is IgM. So let's go through the option one by one. First option, IgA. IgA provides local mucosal immunity. Regarding option B, IgG. IgG provides secondary response. What is secondary response? If the body gets exposed to the same antigen repeatedly, then the body mounts an immune response against the same targeted antigen. But the class of antigen on the first exposure was IgM, but on subsequent exposure it becomes IgG. That is, in secondary response, the predominant antibody that is produced is IgG. Regarding option C, IgE, it provides immediate response to allergy. And regarding option D, IgM, which is the correct option, IgM is produced on immediate response to a particular foreign body or antigen, on exposure to a new antigen. So on exposure to a new infection, a new antigen is coming into play. So in this point, body will produce IgM class of antibody. That is, in primary response, the body will produce IgM antibody, not the IgG. Please mind it. Coming to question 6, which of the following complement is the most potent anaphylatoxin? Option A, C3A. Option B, C4A. Option C, C5A. Option D, C6. So, what do you think? What is the answer? The correct answer is option C, that is C5A. Coming to the explanation part, C5A is the most potent anaphylatoxin. It strongly increases vascular permeability. Regarding option A and option B, that is C3A and C4A, these complements are less potent than C5A. And regarding option D, C6 is not an anaphylatoxin at all. So there is no question of considering C6 as anaphylatoxin. Coming to question number 7, chocolate agar is prepared by heating blood to option A 45 degrees centigrade, option B 55 degrees centigrade, option C 75 degrees centigrade, option D 85 degrees centigrade. And the correct answer is option C, 75 degrees centigrade. Coming to the explanation part, both option A and option B, that is 45 degrees centigrade and 55 degrees centigrade, are too low for RBC lysis, whereas the optimum temperature required for RBC lysis is 75 degrees centigrade. If you heat the media at 75 degrees centigrade, then it lyses the RBC and liberates two growth factors like factor 10 and factor 5, which is extremely essential for growth of Haemophilus influenzae, which is a nutritionally fastidious organism. If the temperature required is 75 degrees centigrade, and you can easily rise RBC at 75 degrees centigrade, then why we will elevate the temperature unnecessarily to 85 degrees centigrade? That is option D, which is more than the required temperature for RBC lysis. So there is no justification of heating the blood at 85 degrees centigrade when the target can easily be achieved at 75 degrees centigrade. Coming to question 8, transformation in bacteria refers to option A, uptake of naked DNA, option B, DNA transfer via bacteriophage, option C, DNA transfer via sex pillars, and option D, random DNA recombination. So what will be the answer? The answer is option A, uptake of naked DNA. Coming to the explanation part, uptake of naked DNA is known as transformation. Option B, DNA transfer via bacteriophage. This is the modus operandi of transduction. DNA transfer via sex pillars. This process is known as conjugation. And random DNA recombination. This is a very vague option. There is no defined method of reproduction of bacteria which counts random DNA recombination as its mechanism. Coming to question number 9, facultative anaerobic bacteria means option A, bacteria that cannot survive in oxygen, option B, prefers oxygen but grows without it, option C, requires oxygen, option D, grows only under high CO2 concentration. Guess the right answer. The right option is option B, prefers oxygen but grows without it. Now let's consider the options one by one. So let's consider the options one by one. Coming to option A, bacteria that cannot survive in oxygen. These are known as obligate anaerobes. Why? Because they cannot tolerate oxygen at all. They require strict anaerobic conditions for their survival and growth. That is why they are known as obligate anaerobes. Option B, prefers oxygen but grows without it. These are known as facultative anaerobes. Why? Because they grow both in presence of oxygen and without presence of oxygen, in absence of oxygen. Okay, so they are not strictly anaerobes, they are facultative anaerobes. Option C, requires oxygen for their growth. As these organisms can't grow without any oxygen in the environment, and they are unable to survive also in absence of oxygen, so they are known as obligate aerobes. Okay, option D, grows only under high CO2 concentration. These type of organisms are known as capnophilic organism. Capno means CO2. CO2 philic means loving. So CO2 loving organism, capnophilic organism, such as pneumococcus. They require 5% CO2 for their growth. Okay, so hope you can understand why the option facultative anaerobes is the correct option. Coming to the last question of this presentation, lipid A of lipopolysaccharide is responsible for option A antigenicity, option B endotoxic shock, option C opsonization, option D capsule formation. The correct answer is endotoxic shock. Let's discuss the options one by one. Option A antigenicity. Antigenicity is not determined by the lipopolysaccharide but determined by the O antigen of the gram-negative bacterial cell wall. Option B endotoxic shock. It is endotoxic shock means the shock like symptoms and signs that are produced by endotoxin. Where do we get endotoxin? The bacterial gram-negative bacterial cell wall produces endotoxin from the component of lipid A. Lipid A is present in the cell wall of gram-negative bacteria, isn't it? So that lipid A portion of the cell wall of gram-negative bacteria is responsible for liberating endotoxin, and this endotoxin produces endotoxic shock. Option C opsonization. What is opsonization? It is the engulfing of the organism by our immune system. So in opsonization there is no role of bacterial cell wall. It is the entire role of our immune system, our antibodies, okay, which is responsible for the opsonization. Option D capsule formation. Capsule is a separate entity. In some bacteria there is an outer covering external to the cell wall. This is known as capsule. Capsule production is not related to cell wall structure of any bacteria. Okay, hope you enjoyed my presentation. If you have liked my presentation come test series, please do like, share and subscribe my channel so that I get more motivation to prepare such presentation and enrich your knowledge. Thank you.